Hi, thank you for visiting my post deciphering the underlying dynamics of cell and gene expression during injury using unedited single cell data. I am Ranojoy Chatterjee from George Washington University from the Department of Biostatistics and Bioinformatics, and I will be the presenter of this poster. During injury, a plethora of changes happens inside and around the wounded area. Body's first response is to close the affected site to stop the infection from growing and also kickstart the recovery process. Very little is known to researchers about how the initial process interact with each other to facilitate this healing process. In our work, the main motivation were to unearth the alterations of cell type identity happening due to the change in underlying biological state. And for us, this change is injury. We also wanted to see the differentially expressed genes between the normal and the injured samples and infer how the recovery process are taking place and which genes are the driving factors for this undertaking. Secondarily, we also wanted to see the difference in trajectory between a chosen cell type amongst the two categories. And finally, we wanted to infer some cell to cell interaction that are happening, which will highlight how different cell types interact and change among each other. It also gives us a detailed network of this complicated yet dynamic network. As you can see in the integration of samples, we use Surat functionality to integrate the 4 mice dataset that we have, 2 coming from normal slash uninjured mice and 2 from injured mice. The integration was done based on anchor genes where the injured samples were taken as reference and the injured, where the uninjured sample was taken as the reference and the injured data was the queried set. You can clearly see from the plot that very few cells were mapped from the queried to the reference. This behavior is expected since during injury, the upregulation of inflammatory cells masks the true identity of some of the cells and thus becomes a very hard job for integration. Based on the result, we decided to break down our study into 2 broad sections, matched and unmatched. Matched refers to the set of cells which were successfully integrated using anchor genes, and the unmatched are the cells which were not integrated. In this plot of integration of samples, the points are cells. We also ran 2 divers to see differentially expressed genes between the 2 categories using the both strategies. This differentially expressed gene is seen on the plot on the top right. We see the differential expressed genes from fibroblasts. We show the differentially expressed genes from fibroblasts, macrophages, and stroma. The top 20 differentially expressed genes was used as the reference, and based on this top 20 differentially expressed genes from fibroblasts, we mapped macrophages and stroma. In the bottom section of the plot, we showed how the difference between the most significant genes and the difference between expression in injured and uninjured category. Similarly, we also ran a differentially expressed test on unmatched data and followed the same strategy. In the trajectory analysis, we emphasize on the different cell state that is existing under certain cell type. This analysis shows that if there is a difference between the 2 groups based on cell state, it's highlighted. In our study, we subsetted the fibroblast cell types from the injured and the normal sample and ran a trajectory analysis. The right-hand cluster comprises of all the injured sample and some normal samples, and the left-hand cluster consists of solely normal samples. Here we distinctly observe how there is a change in the cell state between the 2 categories. Also, how far the injured sample is from the normal sample based on pseudotime is also visible clearly. We also see many nodes in the normal state, which identifies end state, but that is completely devoid or missing in our injured sample. This phenomena is due to the change in the biological situation cause for wound. To visualize the interactions that exist between intra and inter cell types, we use cell chat tool available in R. The comprehensive network brings to light how various cell types communicate within themselves and with other cell types. In the normal tissue sample, we see a lot of interaction, but as we move to the injured sample, we see very few interactions. To conclude my presentation, I will try to highlight some of the key points in the study. Firstly, we try to pull out some of the important genes that influences this process of healing. And also, we try to show the difference in interaction and trajectory that occurs with the shift in biological state. Again, once again, thank you for visiting my poster. Hope you have a good day. Bye.